For today, we'll be going over two chapters, chapters 15 and 16. Chapter 15 deals with the evolution of microbial life, so we'll look at how life began here on Earth and how it then evolved into microbes and into archaeans and then into protista. And then we'll be looking in chapter 16, which is looking at life as it evolved into plants and fungi. For us to better understand how microbial life or how life began on Earth, we have to think back to how early scientists thought about life and how life came about. So for the longest time, scientists believed in this theory called spontaneous generation. And this was the idea that life can come about from non-living material. So we know that, if we want to have another human, then you have to have two humans come together, engage in sexual reproduction to create offspring. So that would not be this definition. But there was plenty of times where in the old scientific world, they would have examples where they feel like life just came from nothing or from a non-living material. So let's say like a piece of meat all of a sudden would have maggots start growing on it. Without realizing that probably flies came in and laid eggs on that maggots. And so there was this big, in the early scientific community, this idea that life can come from non-living life. Well, in 1862, my favorite scientist, Louis Pasteur, he is the guy that invented the idea of pasteurizing milk. That's why we call it pasteurizing from his last name. He's also created our first early vaccines. He also helped with understanding why wine is able to ferment and all that kind of stuff. So he's a pretty big guy in this scientific community, specifically in the microbiological scientific community. And one of the things he did is he proposed this idea called biogenesis. And biogenesis is this idea that life can only come from life. And he wanted to prove this, so he designed an experiment. Because him being a microbiologist, he used a broth that would grow microbes in it, and he would leave it out into the open, and then he would have another broth and he would put a lid on it. And he let them sit for about a week, and when he came back a week later, in the broth that was open up into the environment had a bunch of microbes growing in it, where the one that was closed in the environment didn't. And so he was trying to show scientists that something must have come in here for these bacteria to grow. Well, a lot of scientists then said, well, how do we know that you just didn't cut off the oxygen supply needed and it killed anything that tried to grow in here? And so he then redid his experiment. Same idea. Kept one that was open like this, and then he created something called a swan neck flask. And this swan neck flask would attach onto this flask here, and it allowed for air to come in and out. But he hypothesized that whatever bacteria that were in the air here would not be able to traverse this tube to seed life in here. And so he left both of these open for a week. And once again, this one was full of bacteria and this one had none. So he was able to show, okay, I let air come in here now, and once again, no life grew in here because nothing was seeding the life previously. And so this was in 1862. when this idea now of biogenesis was taking hold. And that this is kind of what we believe throughout science now, is that everything comes from something. We can't just have something just randomly come out of anything. Well then, when you now think of the bigger question, for thinking of if life must come from life, how did life begin here on Earth? And so there are three main theories to this, and so we're going to talk about that. All right, so the first one we'll get out of the way, and that is from a higher power. Right? Some almighty being, some God, some deity, whatever you want to look at it, created life here on Earth. That's a viable hypothesis. Second would be life caught a ride on either a meteor, asteroid, or comet to Earth. So in other words, we know that our Earth in the past had been struck by meteors, asteroids, and comets, and there could have been microbial life living on those meteor, asteroids, and comets, and that survived the impact when it hit Earth, and then it was able to, in a sense, seed Earth with life. All right, so the third one is one that we call chemical evolution. And chemical evolution is referring to this idea that the environment was just right within pre-mortal life, so the pre-mortal Earth, Earth before life, that the chemical arrangements were just a certain way that life was able to start from a series of events that would happen that would allow life to then begin. Okay, so those are our three options when it comes to how life began here on Earth. Now, the first two options, why viable, they're not really, scientists can't go further into them, right? Because they don't ultimately answer the main question is, how did life begin in general, right? Because even if there was a higher power, well, how did that higher power get life? If there was a meteor, asteroid, or comet that brought life from another planet or another part of the universe, well, how did that life begin? And so for us as biologists and scientists, this is the one we study the most, is this chemical evolution. This idea that maybe life began by the evolution of these chemical particles coming together to then seed life. And that's what we're going to be talking about here. What would be the 4 steps needed for life to begin on Earth? To understand how chemical evolution could occur, there's 4 steps that scientists broke down that kind of lead to this idea that we could create life through this chemical evolutionary scenario. So the first step is the abiotic synthesis of organic molecules. So abiotic is without life. So in other words, using things that aren't living to create organic monomers. So remember, we talked about organic monomers. These were things like our sugars, these were like our nucleic acids, our amino acids, those type of very simple organic molecules needed for life. And so in the mid-1900s, we had a group of scientists, Miller and Urey, that came up with this primordial soup idea. And this idea is, okay, we know roughly 4.5 to 5 billion years ago, the environment of our Earth. It was very warm, very hot, and it had a very heavy atmosphere. And so what they did is they created a contraption to mimic the primordial soup or the oceans of early Earth, and to see if they mimicked those different aspects of the environment on Earth, if you could form these organic molecules. And so there were certain like basic elements that were found in the water and this primordial soup. And what they did is they heated this up, and so that it would evaporate these molecules and the water into our early Earth atmosphere. And then in our earlier atmosphere, because it was such a heavy atmosphere, there was a lot more lightning strikes than we see today. There was all these lightning strikes happening. And so they put in this wire here to be able to spark and create kind of like simulated lightning. And the whole idea here is by adding energy here to evaporate our molecules, and then energy here. So we talked about trying to get elements to come together to create molecules requires energy. Otherwise it takes forever for a long time for them to find each other. So the electricity that would spawn from this heavy atmosphere would provide some of those energy. And then what we would do is they ran cold water here through here to help condense those gases so that they would then run back down into the soup and heat back up and go through this whole process over and over again. And what they did is they would let this run for about a week, sometimes up to a month, and then they would siphon off some of this material that had been going through this loop to see if there was any organic monomers that came about. And so they did. They let this run, and when they siphoned off, they found, yes, in fact, they were able to have organic molecules come about through this process. So we know that just on life in that time, because of its environment, that organic molecules would be available and would be made. That's only step one. So step two is now we got to get those organic monomers, right, into polymers. So we have to get those starting to come together. Once again, that's something that's going to have to take energy or whatnot to be able to get those to come together. So another group came up with this idea of like tide pools. So if you've ever been to like the coast and you go to tide pools, so what happens is during high tide, the water comes up way high, and then on low tide, it comes down. And what will happen is some of the water gets stuck in these little pools, because maybe you're not on a beach, maybe you're in a rocky area, and they create these little tide pools where things get stuck in, and sometimes you get like crabs that get stuck in there, fish that get stuck in there, all that kind of stuff. Well, some scientists hypothesize, what if a similar thing was happening? So here in our primordial soup, we know we're making these small organic monomers. What happens if we were to create another type of a thing where you had this platform with kind of these holes in it to represent tide pools, and you would take this primordial soup and you would splash it onto here, kind of like a tide coming in, which would allow these monomers to now be in these small little pools together. If you remember back in our experiment, we talked about trying to get things to come together. We talked about how we wanted to get one male and one female to come together to shake hands. But what happens if you brought and how long that would take for them to find each other blindfolded. But what happens if you brought 20 males and 20 females and told each other to come and shake hands? And that's the idea here, is that we're adding more of these monomers together in a smaller area through these tide pools, where they could actually come together and be able to create polymers. And so they ran this experiment for a while as well, where they just washed on this primordial soup back and forth, off and onto these tide pools. And sure enough, what they found after doing that for many days and up to a month, that yes, they were able to create polymers through this process. So once again, now these first two things have been able to shown to happen. And then the next would be the formation of a pre-cell. Well, the cool thing is we've already kind of talked about this with our phospholipid bilayers, right? If you have those phospholipid bilayers and you put them in a water environment, and you have one end that is hydrophobic and the other end that's hydrophilic, then what happens is they arrange themselves in these circles. This is very badly drawn. This ought to be very small. But they arrange themselves in these circles with the tails in the middle and the heads on the outside and the heads on the inside here. And so they make these kind of pre-cells. And what they noticed is as they ran this experiment long enough, that eventually you ended up making some of these phospholipids, and they started combining with each other in these pre-cells. Not only that, but what they found out is when they made these pre-cells, some of these monomers and polymers ended up inside these pre-cells. And so here you have this pretty much this nice formation of a very prototype of what a cell would look like, the first living organism. Which gets us to our fourth step here. So now you have this pseudo cell. How does it start to replicate, this self-replication? Because this is the most important part of beginning life here on Earth, right? Because if you could start life, but then it never replicates into more life, it's going to die and it's going to be extinct. And who knows, that could have happened hundreds of thousands of millions of times on Earth before your first life was able to start to self-replicate, and then that is the beginning of the evolutionary history of life on our Earth, right? And so this origin of self-replication is the most important part. It's the only part right now in science that we don't have that exact answer for. They're still working on this. They're still trying to think, what are ways that this could happen? And remember, this probably happened over thousands, millions, if not billions of years in our oceans before this first started to happen. So whatever that unique and magic situation is, we don't know. It's going to be really hard to find out. But one thing that a lot of scientists are thinking is that RNA is the biggest factor in here. Just because we see how much RNA connects proteins and DNA and all these other things. So a lot of scientists think that RNA is probably the individual polymer that starts this whole process of self-replication. But currently, like I said, we haven't figured this one out. This is the area in science that we're still looking to figure out. Yes, is this how life began on Earth? Is it through this process? All right, so we do know that life did begin here on Earth, and we can trace that to about 4.6 billion years ago when the first life showed up on Earth, whether it came from a meteor or came from God or came through this chemical evolution. What we're going to do now is look at the evolution of life from that one single molecule. So that single molecule that we say is kind of microbial or bacteria in form. And so we're going to be talking about bacteria and how it is. And then we're going to go to the next advanced life, which is archaeans, and how they are and what they do. And then from archaeans, we're going to go to protista, which is our first eukaryotic cells. That's when things get a little bit more complicated. So we'll look at different eukaryote. And then from there, we're going to go on to plants, which is the next level of development in evolutionary history. Then from plants, it was fungi, which was the next level. And then from fungi to be animals. And animals will be chapter 17. So we'll spend all of chapter 17 talking about animals. Now that we know some ideas on how life began here on Earth, let's look at that life, and starting with our microbial life. Bacteria. So what do you know about bacteria? All right, so you're probably thinking to yourself, makes me sick. That's usually one of the first things people think about when they think of bacteria, is that it makes them sick. So we got a video we want to watch.
Hey, sometimes there are little things that we can't see, and sometimes those things can be kind of scary. We have to be careful. Hey, look, there's Broby. Let's see what Broby is up to.
Oh, I forgot. I'm still hungry. Yummy, yummy.
Stop! 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 Don't eat me! Don't eat me! I'm dirty!
No! Broby! When food gets dirty, it gets covered with tiny, ugly germs. Look!
We are the tiny ugly germs! We are the tiny ugly germs!
They are dirty. And they stick.
We will make you sick! We are the tiny ugly germs! We are the tiny ugly germs!
The things you find on the ground. Don't put them in your mouth. Like coins and fries and bits of food. Don't put them in your mouth.
We are the tiny ugly germs! We are the tiny ugly germs! When the germs are on me, don't eat me, you'll get sick. Those tiny ugly germs can make you sick. We are the tiny ugly germs! We are the tiny ugly germs! We are the tiny ugly germs that will make you sick!
I'm sorry, Bread.
It's okay, Broby. These tiny ugly germs will make you sick.
So just like we see there with our friends DJ Lex and Broby and our robot friend, germs can make us sick, or bacteria can make us sick. And that is definitely the case, but there's a lot more to bacteria than just that. So most bacteria actually are not pathogenic, which means they don't actually make us sick. The reason why we learn about the ones that do is because that's what affects us. And so you hear about those more often than the other ones. But the majority of our bacteria do not. Roughly 40 million bacteria can be found in a gram of soil. So if you were to just take up a gram of soil, you're gonna have 40 million bacteria there. So they're kind of everywhere. Bacteria has the body mass more of animals than actually animals and plants put together. So in other words, the idea here is if you were to take every animal and plant and put them on a scale on Earth, and then take every bacteria and put them on a scale, bacteria would outweigh all animals and plants. So it kind of gives you an idea of how much bacteria is out there. And even within us, if you count up all of our cells that are in our body, roughly in our body there's 10 times more bacteria cells than there are our own cells in our body. So they actually play a very important part in who we are. They help us in our immune system, they help us with digestion, a bunch of that kind of stuff. It's just that those bad ones give them all a bad name, those pathogenic bacteria. So speaking of our pathogenic bacteria, we have a few different ones. There's two types of pathogenic. There's either ones that have endotoxins or ones that have exotoxins. So endotoxins is that they have the toxins that are inside their cell, and so when they attach to another cell, that's when you get that sickness. Or exotoxins, these are ones that actually release out the toxins into the environment. All right, so some that have endotoxins like salmonella, you've heard about that, getting salmonella poisoning if you're eating eggs that have salmonella bacteria in them. Or rickettsia, so this is Rocky Mountain spotted fever that comes from a tick, but the tick actually has a bacteria in its saliva that causes that disease. Some of our exotoxins, so Staphylococcus aureus, so a staph infection, you've probably heard of that before. It kind of shows this red thing on your arm. That's because of an exotoxin. And then lastly, Clostridium botulinum. This is actually the bacteria toxins we use for Botox. So Clostridium botulinum creates the most deadly endotoxins known to man. And what did we do? We created a beauty product out of it. Because what it does is it kills the ends of your nerve endings and stops them from being able to pull on your skin. So if you were to get, Clostridium botulinum in your body, it would kill all these different nerve endings and you'd eventually die. But what they do for Botox is they take some of that exotoxin, they put it in this syringe here, and they put it in specific areas to kill off the nerve endings of the ones that are pulling on your skin to create those wrinkles. And that's how Botox ends up working. All right, so after bacteria, we have archaeans. So archaeans are very similar to bacteria, but they're also similar to eukarya. So we have some similarities here. So we have, they have similar cell walls as bacteria, alternative types of metabolism like bacteria, but then they also have a more complex polymerase chain, RNA polymerase, and an unmodified tRNA similar to eukaryotes. So some ideas of some different archaea. Whoops. Yep. So here are some of our different archaeans. We call them methagenophiles, extremophiles, and thermophiles, hyperthermophiles. So our methanogens, not methians, sorry, methanogens, these are ones that actually produce methane. So methane is that nice gas that you smell. In fact, these archaeans live inside our intestinal systems, and they help break down food products. And part of that, byproduct of that, is methane gas, and that's what causes us to pass gas and have flatulence. So these are actually mud bogs. So if you've ever been to a mud bog, when they boil up the methane that's been created from the archaeans that live in there, and it makes it smell really, really bad. Then we have our extremophiles, these ones that live in extreme environments. So this is actually the Great Salt Lake. We know the Great Salt Lake is extremely salty, and a lot of things can't live in there, but these archaeans can live in there. And then we have our hypothermophiles, so these ones that live in really hot temperature areas. So here is at Yellowstone, one of those hot pools, and you see all these different colors. Those different colors are actually the archaeans that live inside that really hot pool area. And then this is a vent in the middle of the ocean, and it's really, really, really hot. There's also archaeans that live there as well. All right, so let's talk a little bit about Protista. So Protista is our first eukaryotes. And the interesting thing about Protista is we have Protista is this group down here. It's kind of what we call a garbage pail. There's a whole bunch of different types of Protista. And these different types of Protista, some can be similar to plants, some can be similar to fungi, and others are similar to animals. Because the way we determine whether or not you're a plant, fungi, or animal is based on how you get your nutrients. So plants use photosynthesis. Fungi, they actually decompose and break down and then absorb their nutrients. And then animals consume and eat their nutrients. And so there's some protists that do photosynthesis similar to plants, that move around and grow similar to fungi, and others that consume their food similar to animals, but they don't have all the other aspects that makes them those things. So we throw them into this Protista group, where there's other organisms that don't do any of those as well. All right, so here's some different Protistas. So we have our protozoans. So this one here is Plasmodium vivax. It's this little guy here. This is our blood cells, actually. And Plasmodium vivax actually creates a very well-known disease, malaria, that's carried by mosquitoes. So this Protista lives in the saliva of these mosquitoes, and so when they bite you, that's when they can infect you. And you can see kind of the life cycle of this. So this protozoan has to live both not only in mosquitoes, but also in humans. And so this is the whole life cycle of malaria, of the disease of malaria that's caused by Plasmodium vivax. All right, now we have slime molds. So these slime molds act very similar to fungi, where they can be in their life cycle, they can be these singular cellular things. Then they can combine together to create the slime molds. This is actually a picture of one. And then when they want to reproduce, they go into different, they differentiate into different areas of the slime mold to then create this stalk that then creates new slime mold cells. All right, then here we have a paramecium. So this paramecium is very similar to to animals in a sense, because this is kind of like a mouth opening, and it actually siphons. And we've got a video that shows this. Our next is our unicellular and colonial algae. So these are different types of algae. Here we have our diatoms, we have our volvox, we have our pedestrium, and then we have our dinoflagellates. So the interesting thing about some of these is you can see they're starting to work in groups here, and this is the idea of going from single cellular organisms to then multicellular organisms. And there's kind of a cool video here with our dinoflagellates, this bioluminescence that occurs by them.
All right, science experiment, take one. Make sure you point it at the water.
No, no.
So it's pretty cool. By disrupting those dinoflagellates, they actually release this bioluminescence light on them. All right, last is our seaweed here. So we have our blue-green algae, we have our red algae, and our kelp. So you probably thought these were actually plants, but they're not. They're protista. They're multicellular protista that are very similar to plants, that they do photosynthesis and other types of things that are similar to plants, but then they also lack some of the other things that are more specific to plants. And then next we're going to be looking at plants and fungi. So really quick, for chapter 6, we're going to go over. So really quick, for chapter 6, we're going to go over plants and fungi and the move to land. So our first types of plants are we have 4 types of plants. We have our bryophytes, our ferns, our gymnosperms, and our angiosperms. And we're going to look at our bryophytes. And so when life was in water, in the ocean, it was able to always be around water, and it was always able to do anything it needed. It didn't have to worry about water. So once life went to land in the form of plants here, we see that now there's this issue with having to deal with water. And our very first evolutionary types of plants are our bryophytes. They're very small. They're our hornworts, our liverworts, and our mosses. And so they're very small, and you'll find them in very wet environments. And the reason for this is because they rely on water. They rely on water to be able to help them to not only to live, to have that water, but also to help them to do sexual reproduction as well. From there, we go on to our non-vascular and seedless plants. And so here's our bryophyte's life cycle. And you see that they actually use water. So we have these 2 different types of spores that come about. One is the gametophore, one is the pronindium. And they create 2 different types. One's a male, one's a female. And the male has these sperm that can only travel with water. And so that's why you only find these bryophytes in very wet environments, or they wouldn't be able to do sexual reproduction. Our next group is our ferns. Ferns are also tied to water. They're vascular and seedless plants. They still don't have seeds, but they're also tied to water. That's why you see ferns in very wet environments, but they're a little bit bigger. They now have a vascular system. So our other plants didn't have a vascular system, which is how they get water to them. So we talked about this on cohesion and adhesion with water and the utilizing of the xylem and phloem. So the xylem allows water to flow from the bottom, so the roots, all the way up to the shoots or the leaves and the branches, and it goes in this up direction. The phloem is vascular tubes that go the other way, and they get all the nutrients. So remember, our leaves and stuff like that are the ones that create our sugars because they do photosynthesis, so they can share all those sugars and nutrients back down to other parts of the plant. And so think of these as kind of like the arteries and the veins of a plant. That's their vascular tissue, is the xylem and phloem. Now here you see the life cycle of our ferns. Looks kind of similar to the life cycle of our mosses, where they have these spores and they have a gametophyte and an antheridium, and so they have a male and female parts here. And once again, the sperm is needed to go in the water to be able to get from the antheridium to the archegonium to fertilize the egg, and then you create your fern, and that's that kind of life cycle. Next, we're going to have our vascular and seed bearing. So these are our plants with seeds. We first have our gymnosperms, and then we have our angiosperms. So our gymnosperms, they are the ones that kind of have more like cones and stuff like that. And these are some of the more non-traditional gymnosperms. So these are ginkgos, our cycadades. All right, so these are our conifers. And what we notice here is that our conifers, you have cones. And we have our female cone and our male cone. You probably didn't realize that there's two different types of cones. So our female cone here houses the ovium and whatnot that needs to be fertilized. And then our male cones are these right here. And if you ever grab these, if you've ever been out in the woods and you grab these, they kind of flake off. Well, this is actually the sperm that goes into, or the pollen that goes into the cone to fertilize it. So this is an x-ray here of that. So here those little pollen come down and they fertilize these eggs that are here in the male cone. All right, next we have our angiosperms. Our angiosperms are our flowering plants. So in our angiosperms, our flowers, we have male and female parts. So we have our male parts, which is the stamen that has the filament and the anther, and this is where you have your pollen. And then the female parts, which is carpel, sometimes called the pistil. We have the ovary, the style, and the stigma. And what happens is these pollen here will go down the stigma, down the style, don't need to zoom in, and then fertilize the ovium down there to then create the seed that is needed to that flower to then be replanted. So why are so many angiosperms colorful and fragrant? Why do they smell so good and they have these beautiful colors? Well, the whole idea here is that if you go back to this picture here, it's really hard to get the wind to be able to drive this up in there. And that's what happens with our conifers, is all that, when you break all that, the wind blows it around and that gets in there. Well, it's hard to do that here. So instead we have insects or other types of animals that will come in and collect the pollen and go from flower to flower, and eventually pretty much is the conduit for sexual reproduction for these plants, as it brings pollen from one flower to another flower, and the pollen gets stuck on the bee or the moth or the butterfly's leg, and then it gets down into the pistil, and that's how it's able to have fertilization of the ovum. All right, there are two different types of our flowering. We have our monocots and our dicots, and these are different ways to determine which they are. So some of the easier ones to recognize is how their leaves look. If they're kind of spider leaves like this, or then they're a dicot. If they're more linear, then they're a monocot. And then the really easy way to look is on their flower petals. If their flower petals are multiples of 3s, so 3, 6, and 9s, then it's a monocot. If it's multiples of 4s and 5s, then it's a dicot. Also, another easy way is by their root. If they're a fibrous root system, so they spread out everywhere, then that's a monocot. If it has a nice big tap root, so think of like a carrot is a tap root, then that would be a dicot. All right, now let's talk about our fungi. So here's some different pictures of different fungi. And that guy as well, he's a fun guy. All right, so how fungi work is they have two different parts. So most of the time our fungus look like this. It's what we call our mycelium mass, and you don't see them very often. This is kind of their normal life cycle is in this mycelium mass. When they want to reproduce, that's when they create this fruit body, which this would be a fungus, right? And are like a mushroom. And underneath the mushroom here, are these pores where they then send out their different spores to create more mushrooms. And so you only really see mushrooms, or more fungi I should say, you only really see mushrooms when they're looking to reproduce. Other than that, it's all over. And so here's this unique little idea here called a fairy rings. So these, what happened is overnight you would have these mushrooms that would grow overnight. People wake up and they're like, oh my gosh, what happened? And so there was this fairy tale that people would tell their kids that that fairies at night would dance around in circles, and everywhere they touched, a mushroom would grow up. And so they called these fairy wings. What actually was happening is this mycelium mass had grown so far, and it was used up all of its area, so now it wants to be able to reproduce, and since it grows kind of in a circular format, it then reproduces on the edges of that, and that's where those mushrooms pop up. So really that fungus lives all over in this grassy area right here, and then at night those popped up because it's looking to reproduce, because it's running out of area, so it's trying to set out its spores to go find other areas to grow. All right, we use fungi in food quite a bit, so whether it's eating them, looking at like our portobello mushrooms, or if it's creating breads, cheeses, and beer. So we talked about how we use fungus to do this before when we looked at anaerobic respiration. Anaerobic respiration, where we saw when we took fungus, we were able to use yeast, which is a type of fungus, to create the alcohol in beer and the bubbles in beer. And you also can do that to help bread rise, because that CO2 that releases from that helps the bread rise. And then also it's utilized in fermenting some cheeses as well. Another really cool thing about fungus is that they can also create antibiotics. So Alexander Fleming in 1928 accidentally discovered penicillin, which came from a fungus called Penicillium. And what he did is he was working on all these different types of bacteria projects, and he had a plated bacteria. So here's this plated bacteria, and he accidentally left the plate out. And then the next morning he came, and a fungal spore had landed here, and he saw everywhere where the fungal spore was growing that it was inhibiting the growth of the bacteria. So he learned more about it, went to find out, and he found out that that fungus was actually releasing these toxins that would kill the bacteria. And those toxins are antibiotics. And so a lot of our antibiotics come from different types of fungal strains. We also have antibiotics that come from bacteria as well, because bacteria are competing with each other, so they create antibiotics. So those endotoxins that we talked about, or the exotoxins that we talked about. And then even today, we now, because we know what to look for, we can actually manipulate and create our own antibiotics, in a lab and whatnot as well. All right, that is the end of these two chapters. We'll start on chapter 17 next, which will be looking at animals.